Hello everyone and welcome to Lab Manager's Tech Trends webinar series. My name is Trevor Henderson, Technology Editor for Lab Manager, and I'll be moderating today's discussion which will focus on cell culture techniques. Today our panel of industry specialists will discuss methods to automate and streamline your cell culture processes and laboratory workflow. We like our webinars to be very interactive, so we encourage you to send us your questions at any point during the webinar, and our panelists will discuss these during the question and answer session following the presentations. To ask questions, simply type your query into the question box located at the bottom left-hand side of your screen. We will try to address as many of these questions as possible during the Q&A session. However, should we happen to run out of time, I will forward any unanswered questions to the panelists and they can respond to you directly. Additional resources can be accessed through the resource widget located at the bottom of your screen. You may also move or resize any of the windows simply by grabbing them at the top or stretching them at the bottom right corner. You may also have to move or adjust the windows to make the live view visible. This webinar recording will be available early next week on Lab Manager's website. At the end of this webinar, we'll share that link with you as well as the contact information for all of the panelists. With that, let me introduce our first speaker who joins us today from BioTek Instruments. Peter Brescia, Jr. is an application scientist at BioTek. He holds a master's degree from UVM in cellular and molecular biology and an MBA in performance measurements from Champlain College. Peter's current work involves automation of microplate-based assays in the areas of epigenetics and stem cell biology. Thank you for joining us today, Peter.
Thank you, Trevor, and thank you all for attending today. Today I'll be discussing cell culture techniques that include automated cell dispensing and image-based monitoring of spheroid formation. These are recent methods that were developed to help aid in the generation of spheroids and the use of automated digital fluorescence microscopy to aid in the tracking of spheroid formation in a microplate-based format. I'll include a brief introduction today to 3D cell culture, as well as the instrumentation and materials used, and an application note that I'll present, and I'll describe in more details the workflows including cell seeding, media exchanges, and automated image-based monitoring. Any additional information regarding the instrument specification and methods not covered in the presentation can be found by following the links included in the presentation resources section, or they're also available at biotek.com. So just a brief introduction on 2D versus 3D tissue culture. The primary reason for the exorbitant costs of new drug development is due to the extremely high, approximate 95% failure rate that's attributed to either lack of efficacy or unforeseen toxicology at later stages of drug development. And much of a candidate drug's early discovery and screening has been performed using two-dimensional or cell monolayers that do not truly reflect tissue physiology. So most cost-effective solution is to obtain better targets and initial toxicological results using more relevant cell culture methods before candidates progress to animal testing and further stages of drug discovery. So more recently, new developments in 3D culture provide some affordable solutions that are also amenable to high throughput screening. And these have been found to be better substitute for in vivo modeling or animal testing, as well as the automated processes themselves can provide for more consistent data. So here I've put up a table of some of the available 3D cell culture systems. 3D cell culture systems are available from a variety of vendors, and they can be performed in an array of different formats, from 96 well plates to 384 well plates, as well as your standard tissue culture vessels. Several rely on scaffolds, while others contain no matrix material whatsoever. The application I'll be discussing today, which is highlighted in gray here, is spheroid formation using hanging drop microplates from 3D Biomatrix, and this particular spheroid formation requires no scaffold material. The title of the application note is using the multi-flow FX and citation to dispense cell suspensions into hanging drop plates and to track spheroid formation via digital wide field imaging. And the link at the bottom is to the application note, which again can be found on biotek.com. A brief description of the multi-flow FX, again the additional specifications can be found on the website. Here we go. This is has flexible dispense options, which include two peristaltic pumps seen on the left side of the image. These have low dead volumes and are excellent for dispensing critical reagents and cells. Also, two syringe pumps, which can be seen in the upper right-hand side. Those are for dispensing of bulk reagents. Also available are cell-friendly dispense and wash tubes with adjustable flow rates, which can be used for media exchanges, etc. The small footprint of the instrument, as well as the touchscreen controls, are ideal for use within a laminar flow hood, which is typically where our instrument is housed in our tissue culture facility. And this allows for contamination-free cell dispensing, cell washing, media and reagent additions that are typically required to be contamination-free when performing longer-term assays that might span several days or several weeks. Let me advance to the next slide here. Okay. The Citation 3 is actually 3 instruments in one. It contains the filter-based detection system and monochromator-based systems that are found in in our most recent hybrid systems. We've then combined this, which can be seen in the lower portion of the instrument diagram, with digital wide field microscopy system. And this allows us to capture both quantitative numerical data as well as phenotypic rich data through the imaging. And this gives you a more complete picture of when you're performing cell biology research. So as I mentioned, today we'll be discussing the use of hanging drop plates for spheroid formation. And up at the top left, you can see a diagram of the actual plate, which consists of a lid, the hanging drop plate, and a tray. Both the tray and the hanging drop plate have reservoirs, which can be filled with either sterile water or buffer to help with humidity control. The spheroids themselves are self-assembled microscale cell aggregates. They're generating using the perfected 3D hanging drop plates from 3D Biomatrix and are considered superior models for avascular in vivo microtumors when compared to standard 2D tissue cultures. Using the system, a drop of cell suspension is pipetted, which can be seen in the lower portion of the diagram. The cell suspension forms a stable drop below each well through a small hole in the bottom of each well, where the cells aggregate into spheroids. In both 96 and 384 well plate formats, the spheroids form mass transfer gradients of oxygen, nutrients, waste, and therapeutic drugs that are very similar to what would be observed in tumors within a human body. Two critical steps to the success of generating reproducible spheroids is first, cell and media dispensing into the plate, and second, being able to track the spheroid formation for optimal timing of when to add drugs and such. For cell dispensing, the peristaltic pumps, as mentioned before, were used to add cells of appropriate density to the entire plate, and then the automated imaging was then performed to track spheroid formation over the course of several days or weeks, depending on the cell types used for the spheroid formation. There are several options for assay workflows. The top two here require transfer of the spheroids after formation to an assay plate for any reactions that may require mixing or shaking of reagents once added or compounds. The third method, depicted at the bottom of the diagram, allows the entire assay to be performed in the hanging drop, and this is typically when no mixing or shaking is required, such as when the assay has a colorimetric or fluorometric readout. Spheroid formation typically takes between 3 and 7 days, again dependent on what type of cells are being used or whether it's a co-culture, and drug exposure typically 1 to 3 days. Media exchanges are also required in some instances, and typical exchange regimens include either daily, every other day, or media exchange when a drug addition or compound is required. These are typically accomplished by manual aspiration to remove media. It could be the removal of 10 microliters. Typically, cells are seeded in a volume of 30 to 50 microliters. So removal of 10 microliters and replacement with 14 microliters of fresh media may be required daily or every other day. Typically, you'll have some evaporation as well, so the addition is typically a larger volume. And then just prior to a drug addition, you may want to more thoroughly wash your spheroid, and so we can do multiple removals and replacements of fresh media. Microscopy, as mentioned previously, is used to track formation of spheroids. In this particular example here, MCF7 cells, these are GFP expressing, stably expressing cells. These were added to the plate, approximately 5,000 cells per drop, and were imaged using a 2.5x objective. The cells were being, or prior to being imaged, the reader control, environmental controls were set to 37 degrees Celsius and 5% CO2. The entire plate assembly can be inserted into the reader. The clear tray can be imaged right through that. And the optimum Z height was obtained using manual image mode to determine the height where the largest number of cells were in focus. This can be performed using bright field imaging for unlabeled cells or using fluorescence for labeled cells. And the height can then be used for automated imaging of the entire plate once that is established. This might need to be repeated after day 1 or day 2 as the formation of the aggregate forms. It tends to move to the bottom of the drop, so the Z height may have to be adjusted to get the best images. And again, once you find that Z height in manual mode, it can be set and used for automated imaging of the entire plate. So in the panel here on the right, we can see day 1 through day 4, and we can see dispersed cells in day 1, aggregates forming in day 2, and then certainly by day 4, a much more confluent aggregate formed, or spheroid. And the following examples that I'll show are images obtained while tracking spheroid formation using the hanging drop method when seeded at different cell densities and time points. In the first slide, we have 10,000 cells per well, and this is time 0. Again, these are GFP expressing cells. So on the left side, we see images taken with either a 2x or 4x objective in bright field. In the center are images taken using our green fluorescent channel, and then a composite of the 2 overlaid images can be seen on the right. And we see at time 0, we have dispersed cells. And then if we look again about 15 hours later, see now cells are starting to begin to form aggregates, and spheroid formation is beginning. Media exchange can also be evaluated. On day 1 here we show before on the left side of the slide, and then after 1 exchange we can see we get rid of some of the background and cells that may not have aggregated. And then after 3 exchanges, we see we still have excellent retention of our cells. Next are essentially a time lapse of spheroid development for 5,000 cells per well. Again, day 1 through day 4. We can see we start with dispersed cells in day 1, and by day 4 we have a nice aggregate into a spheroid. Next, we have a comparison of spheroids seeded at different cell densities. Again, spheroid size and shape will be dependent on how many cells were seeded. So here we show cells seeded at 1,000, 5,000, 10,000, and 25,000 cells per hanging drop. And the last image that I wanted to show you is actually a co-culture. Cells were mixed 1-to-1 at the appropriate cell densities and then dispensed as a single dispense step at either 5,000, 1,000, or 2,000 cells per drop. These are a mixture of GFP-expressing MCF7 cells with human dermal fibroblast spheroid fibroblast cells expressing red fluorescent protein. Again, these images were captured using 3 separate channels, bright field, red and green fluorescence, and then overlaid, and we can see we have excellent formation of spheroids. Again, these are, I believe, day 7. And finally, I'd like to just finish up with showing you where you can find more details regarding the information provided today. If you go to the BioTek website, our homepage, and click on the resources, and then applications, hot applications will feature some 3D cell culture application notes, and from there you can download any applicable information that you might need. We also have all of our other application notes broken out by category, so feel free to peruse those. And I'd like to conclude by just making a few remarks regarding the Multiflow FX reagent dispenser. Again, up to 4 independent reagents can be dispensed in parallel, and so this can take care of cell dispensing, media, buffers. The touchscreen control allows it to be placed in a hood for contamination control. And the Citation 3 with the 3 optical paths allows now for fluorescent microscopy as well as your standard optical pathways to obtain both phenotypic and quantitative data. And the recent development of suitable 3D cell culture models, both scaffold-based and non-scaffold-based, are available and amenable to automated high-throughput screening. These recent developments are starting to shift the way folks are looking at drug discovery. And I'd like to thank you all for your time today. And feel free to contact me if you have any questions, and visit our blog, Scientifically Speaking, where we will cover some of our more recent developments. And thank you.
Well, thank you, Peter. And thank you all for sending in your questions. I encourage you to do this throughout the presentations. If you're joining us late, you may simply enter your query into the question box, which is located at the bottom left-hand side of your screen. Our next presenter today is Dr. Kristina Klette, who joins us from Hamilton Robotics. Kristina received her PhD from the University of Nevada, Reno in biochemistry and molecular biology, where she studied cancer cell biology. After receiving her doctorate, she took a postdoctoral position, continuing her research in the cancer biology field, and subsequently joined Hamilton Robotics as a project leader in their custom robotic integration group, soon after becoming the manager of this group. After 3 successful years in the integration group, Kristina became a business unit manager for the Automated Cell Biology Applications Business Unit. In her current position, she focuses on providing researchers with automated solutions for all cell-based assays and managing their cell culture system needs. Thank you for joining us today, Kristina.
Thank you, Trevor, for the introduction. And I would like to say thank you to everybody else who joined this technology webinar today on cell culture techniques. Okay, so to get started, I wanted to give a brief overview of what I was going to talk about. First, I wanted to speak about, why would a researcher or somebody in a lab want to automate cell culture? And then continuing on to talk about the tools that automated systems use in automating cell culture and cell-based assays, and how we tackle the different parts of a process throughout a cell culture process or a cell-based assay. And then I'll show you an example in the end of what a typical automated cell-based or cell culture system would look like. Okay, so as everybody knows, cell biology is a very multidisciplinary field. It's been increasing at an incredible pace. Researchers are now taking a more system approach than just looking at an individual protein or sequence in the DNA and comparing that to protein expression. We're combining all of these different things into one system and a systems approach and looking at all kinds of different interactions of proteins as well as phenotypic characteristics. There's also been a shift in this field to more cell-based assays for, say, drug discovery and compound screening systems. Cell-based systems are more biological applicable than looking at non-cell-based systems such as protein and interactions and as well as DNA expression. So why would one want to automate cell culture? Well, because cell biology is such a disciplinary field, multidisciplinary field, and we have it increasing at an incredible pace, there's this increased demand for the use of live cells with the shift in use of cell-based assays. And with that comes increased throughput needs. So that's why people are looking to automation, because you can't handle this demand anymore manually in a lab. Also, to avoid the variables from one person to the other, labs and researchers are looking to standardize and become more consistent with their protocols, and the robot and automation is able to handle that for a customer. Also, the automated equipment is extremely accurate and precise and will handle the process the same way every time. And maybe one of the most important things is it offers walk-away time. So the researchers can spend more time looking up papers, researching what their next assays are going to be, and, looking for grants, things like that. So we'll let the robot do the mundane day-to-day tasks while we spend our time doing more important things. So I wanted to just point out some of the trends and the most popular applications we see in automation recently. Cell line development is a really big one. We see a lot of pharma companies as well as academic labs looking at expression systems, such as those that produce monoclonal antibodies. Also, iPS work, induced pluripotent stem cell work, is very popular. With any kind of cell-based work, obviously comes maintenance of cells. So we see a lot of interest in automated systems for cell maintenance and culture. Also, with the expression systems and the cell line development systems, we see an increased demand for cryobanking or preserving these cells so that they can be arrayed or genetically characterized later down the road. This is very popular with stem cells. Cell-based assays, as I mentioned before, are extremely popular for drug discovery. Cellular expression systems, as well as there's an increased demand for high content screening for phenotypic changes in a cell, which involve immunostaining procedures. So what does a typical workflow look like in a cell culture cell-based assay lab? And what would the processes look like on an automated system? So typically, you need to start with some cells for your process, and they need to be seated in a plate. Customers are looking to do this on automated systems where they're put into a process that the automated system would handle the growing of the cells, control the incubation of the cells through a period of time that they need to grow, handle the media exchanges on a regular basis, allow passaging of those cells when they become too confluent, or they need to passage them for a cell-based assay. And then to the point where, do we have enough cells for our cell-based assay, or do we need to continue the process through the cell culture maintenance? Endpoints of cell culture and maintenance on an automated system would be doing preparation of assay-ready plates, such as those for differentiation protocols or some kind of cell-based assay for quantitating protein expression, or transferring those cell lines to manufacturing. A typical example of one downstream assay would be an expression development system, where once you've expanded your cell line, you might want to transfect it with some kind of genomic material and place those cells out in, say, semi-solid media for antibody production, and then select the clones that you're interested in based off what kind of expression criteria you're interested in selecting for. Another example would be for IPS development on an automated system. Once you expand your cells, you may want to reprogram them and also passage those and expand the reprogrammed cells that you're interested in and feed those back into a cell maintenance and passaging system. There's a lot of challenges to cell culture, as everybody probably knows, and there's some things that you have to look for when you're looking to automate. One thing is sterility. Obviously, cell culture systems have to be sterile, everything has to be kept clean, and we have to avoid contamination. Also, when you're looking at an automated system, cell maintenance and culture and cell-based assays are long assays, and they run a long time, and looking at the system software stability is an important aspect. Also, keeping your cells in suspension in order to pipette them if you have suspension cells. As we all know, cells tend to settle over time, so that's another challenge that you have to overcome on an automated system, and looking for the right tools to do that job is important. Large volumes of media are handled on a cell maintenance system, especially if you're maintaining cells over the course of 3 weeks. You're going to need a lot of media and a way to apply it to the deck, the instrument, where the pipetter or the liquid handler can handle it. Also, for selection criteria, if you're picking clones, you want to know what type of software and imaging capabilities those systems have, how we're going to separate and fractionate cells that we're interested in that might be expressing a certain protein, as well as any other analytical aspects for imaging and screening. What type of systems can the vendor provide that they can integrate to or provide themselves? Okay, switching gears a little bit, since I gave you all a background, I'd like to talk about the tools that enable automation on an automated cell culture system. So we'll walk through certain parts of the processes of a cell maintenance system and how we tackle those on automation. First of all is the pipetting, obviously, and how we manipulate the cells around the deck. We use air displacement technology, which is very preferable, and it avoids contamination. It's nice because it's very similar to the high-precision electronic handheld pipettes that we're used to using in the cell culture hood. These type of pipetting channels do have a dynamic volume range, and they're extremely accurate and precise. And they're also enabled with pressure and capacitance liquid level sensing. And the nice thing about air displacement is there's no system liquid or self-contained liquid channels, so you don't have to worry about maintaining those on a regular basis, and it reduces contamination risk and also allows for no tubing or pumps. Within the air displacement pipetting channels, there's a technology called core technology. This is a technology used for tip attachment for disposable tips, and it's nice in the fact that it provides a lock and key mechanism. So the pipetting channel actually has an O-ring that will expand into a groove in the disposable tip, and this is the lock and key mechanism I'm talking about. It allows for robust and reproducible tip pickup, so no pressure fit of tips, and increased accuracy and precision of the pipetting. And then when you go to eject tips, it eliminates splashing and creation of aerosols because it gently releases the tip and lets it fall to waste. Also, a nice feature of this is it allows use of core grippers and suction cup tools, which can be used to manipulate labware around the instrument's platform. So here's a couple pictures, examples of different tools core technology can use. We are able to manipulate lids on labware and move those around, and that's really important with cell culture because all of the labware needs to be lidded to keep it sterile, as well as we can move labware around deck with core grippers as well that you see in the lower left. And again, as I mentioned earlier, air displacement channels offer a pretty broad dynamic pipetting range. You can go up to 1 millimeter or even up to 5 milliliters in one pipetting channel. And this allows work with larger culture vessels such as 6 well plates or deep well plates, as well as 50 mil conical tubes. Okay, switching gears a little bit to imaging and colony picking. One thing to consider is the system and how it can handle these types of selection criteria or quantitative analysis. For colony picking, it's important for screening and selection, and we want to see what type of imaging capabilities in terms of the camera that the instrument has to take pictures of all the colonies and how the image analysis software handles the colony selection, whether it be based on cell morphology or color or size, shape, proximity to other colonies, and then how it picks those colonies and aspirates them and transfers them to another labware. Again, as I mentioned earlier, sterility is one of the biggest things to consider. Liquid handling platforms, you can put HEPA filter heads directly on top of the liquid handling platform to allow for a clean air zone and a contamination-free work zone. These are nicely steamly integrated. The other option to consider was putting the liquid handler into a biosafety level 2 cabinet. Companies such as New Air, Baker, Germ Free, and there's many others out there offer very seamlessly integrated biosafety level 2 cabinets that liquid handlers can go into. They also allow for integration of third-party devices or other devices such as incubators or imagers that can be integrated nicely with the liquid handler. Some accessories for the on-deck or the pipetting platform to handle the cells during the culturing process. Refillable reagent reservoirs for sterile media or cell suspensions is an important thing to consider and how those are being supplied to the deck. It's important to look at how it handles SBS labware and what types of formats. Can it handle multiple probe pipetting head? Can it handle independent channels, pipetting channels? And also, does it have a completely disposable sterile fluid path from the source container of media or cell suspension to the instrument's pipetting platform? Also, can that media or cells be stored at 4 degrees or 37 degrees and pumped to the deck via, say, a peristaltic pump and heated to 37 degrees if the media is at 4 degrees for use in cell culture labware and cell plate. Also, when passaging or exchanging media in a cell culture vessel with cells, oftentimes manually in a hood, you manipulate the labware and tilt it so you can remove all the media or exchange it. And we have offered, or other companies, to look at what they have to offer, but in terms of being able to tilt the labware so that we can exchange media and completely remove it out of the labware, or when you're passaging cells, it's important to get all of the cells out of the well. And what type of labware it's compatible with. Also, when passaging cells, especially adherent cells, sometimes they need a little bit of a nudge when you're trying to chemically dissociate them. So looking at what types of devices help allow assisted dissociation. Heater shakers work really nicely for this, especially ones with a very tight orbit of 0.5 millimeters to 3 millimeters, depending on your plate type. And this will allow that little nudge that they need sometimes to lift off the plate so you can passage and harvest the cells. Also, what's nice about having heater shakers on deck is they can be heated without shaking, so if there's a reagent or cells that need to be kept warm while they're being processed, these offer that as a solution. Also, keeping cells in suspension on deck. As we know, cells do tend to settle over time if they're not continually manipulated, and they need to be manipulated gently. So having devices that can mix the cells gently and keep them in suspension while you're plating out, say, hundreds of plates in 384 wells or something like that. So we have the option to have magnetically driven stainless steel paddles that can keep cell suspension going and not settle over time. The other thing is the ability to integrate. As everybody knows, cell culture is a very multidisciplinary field, and along with just maintenance of cells, they need to be quantitated, or some kind of image acquisition has to occur during the process. So looking at how the vendor integrates the devices and what kind of software they offer to do the image quantitation and control of the other devices. So things such as how integrating cytometers, imaging systems such as the BioTek instrument you saw earlier, what types of devices can be integrated in terms of fractionation separation, so magnetic separation devices or centrifuges, as well as incubators is one of the most important things in cell culture processes. So how the incubators handle their inventory and how they integrate with the vendor liquid handler platform. So here's a picture of what a system typically would look like, an integrated cell culture system from Hamilton. You can see here on the right we have integrated incubators. This system supplies about 220 cell culture plates to the system liquid handling platform in batches, where the deck accessories that I mentioned earlier are situated so that we can handle passaging, media exchanges, cell suspension, etc. here underneath the pipetting platform. On top of the liquid handler in the middle, you can see that there's an integrated HEPA filter hood to keep the pipetting surface a clean work zone. And then on the left-hand side of the liquid handling platform, you notice there's a confluence imager and a BioTek reader to do quantitative analysis. And then below the instrument platform, we have refrigerators to keep media cooled, and then it can be heated on the way to the instrument platform to 37 degrees for processing cells, and as well as other control units and modules underneath. And that concludes my talk for today. I'd like to thank you all for joining, and if you have any other additional questions, feel free to visit the website at hamiltonrobotics.com, or you can email any requests to marketingrequests@hamiltonrobotics.com.
Great. Thank you very much, Kristina. Just a reminder that this webinar video will be available early next week on Lab Manager's website. If you'd like a copy of the presenter slides, we suggest that you reach out to them individually via email. Their contact information will be available at the end of the presentation. Our final two speakers join us today from Panasonic. Nehal Kothari is the Strategic Marketing and Application Specialist at Panasonic Healthcare Corporation. Having worked at Panasonic for more than 2 years, she focuses on application-based marketing and strategic research to help in better understanding of product applications and trends. She belongs to a biotechnology background, having secured a distinction for her master's in biotechnology, bioprocessing, and business management from the University of Warwick, UK. Also joining us from Panasonic is Karl Radosevic. Karl is a product specialist at Panasonic. His roles include sales and marketing support, product management, and product development. He earned his undergraduate degree from the University of Illinois with a major in molecular and cellular biology, and completed his master's degree from Northwestern University's Masters of Biotechnology program. Since college, Carl has been heavily involved in various areas of microbiological and biomedical research. Following his studies, he worked as a laboratory manager for a pediatric cancer research lab in Chicago affiliated with Northwestern School of Medicine. Carl also spent several years at Nanosphere, a nanobiotech company in Illinois, where he worked in R&D to develop several molecular diagnostic assays that rapidly determine infectious diseases in clinical specimens. Nehal and Carl, it's a pleasure to have you with us today.
Thank you so much for that, Trevor. Hello, everybody, and thank you for your time today. I'm sorry we had some technical difficulties earlier, but now we're all good and we're all set to take you through the presentation that we have for you today. So Carl and I are going to delve into a topic that is crucial yet mostly ignored in a laboratory environment. There have been references that prove everything in this world behaves like a biological system. Simple law of physics as well. There has to be a balance in the universe, right? If one component increases, it has to be compensated through different ways to allow for that increase. Such state of homeostasis, you may say, is essential even in a laboratory environment. Although we can go on and on about how this holds true in every scenario, we are going to focus more on the growth environment for cell culture, especially in an incubator. Moving on, just as an introduction, we thought it would be interesting to see what are the current market trends in the cell culture industry. It is no surprise that primary cells, as you can see from this graph, are the most widely used cells in the industry, followed by continuous cell lines. The main reason for this was attributed by this research to the fact that primary cell cultures are more closely mimic the physiological state of cells in vivo and therefore generate more relevant data compared to the transformed continuous cell lines. Because of this, the primary cells are most widely used to understand the in vivo model system as well as for drug discovery and screening processes as seen from the second graph. Use of continuous cell lines is more within the ADME and the toxicology studies, amongst other things. Further, there are more focused cell lines. An example that we've taken here are the stem cells that mirror the analysis of the primary cell lines. Increased use in the in vivo model system, disease modeling, and drug discovery and therapies is also seen in stem cells. Such stem cell-derived differentiated cells are consistent in nature. However, their downside is, of course, characterization. In recent years, the use of cardiomyocytes have gained popularity in drug toxicity and effectivity screens. However, commercially available ES and iPS cell-derived cardiomyocytes are becoming widely available and accepted as biologically relevant alternatives to current cell models and primary cells for predictive toxicity. Endothelial and epithelial cells, as you can see from this graph, are also expected to increase in the near future. Mesenchymal stem cells have to be mentioned specifically since these are widely used for their multipotency and can be obtained from a number of different tissue sources. They can also be banked for long-term storage and are likely a patient-specific source for a stem cell-based therapy. I feel personally it shows great potential and a very bright future in the near future. The reason that I wanted to mention the trends in the primary and the stem cell-derived cell lines is because both these cell lines come with their own pros and cons. This graph mentions the factors that determine the satisfaction rate of the primary cells. Of course, the price is always a factor associated with any type of cell culture activity. However, cell viability and growth rate require special mention since these are vital to the overall productivity. We will see in a minute how this ties to the homeostasis concept that I was talking about earlier in a minute. Similarly, just an overview for the stem cell-derived cell lines, one of the top concerns in the industrial sector is consistent performance. Knowing that your cells are cultured in the same environment with the same process and media so that it can produce the same results over and over again is one of the major attributes that can be termed as consistent performance. In other words, consistent performance can also be termed as reproducibility. Reproducibility is one component of the precision of a measurement or test method. There are several components that work together to provide a reproducible environment. The cells themselves, as we saw in the previous slide, need to maintain the similar cellular characteristics. Media and consumables used that means having the same media and consumables eliminate the unexpected experimental variation to a certain extent. The process, technology itself, this needs to be extremely thorough for regulatory approvals and GMP protocols as well. And of course, the type of equipment that is used. Technical and or specification changes within the equipment can lead to drastic repercussions in the final result. All these factors need to combine together to provide a reproducible environment, ideal for cell culture. However, it's easier said than done. In 2012, a survey done for Nature found that 47 out of 53 medical research papers on the subject of cancer were irreproducible. We can go into the details of each and every topic mentioned here, but then the conversation will never end. However, we at Panasonic have expertise in the equipment part of this. So let's see in detail how does your equipment play a crucial role in your cell culture setup. We'll be focusing on the cell culture incubator for this particular scenario, as it's one of the most important equipment that can directly affect your cell culture growth. Before I hand over the reins of the remaining presentation to our product specialist, Carl, I wanted to circle back to the first example of homeostasis in an incubator that I spoke about. Going back to the most simple definition of homeostasis, it is the property that maintains the state of a system rather than its functions. This diagram specifically refers to the CO2 environment that your cells are subjected to within the incubator. The external environment that provides carbon dioxide has a direct correlation with the internal conditions that use up the carbon dioxide. The external carbon dioxides within the incubator affect the pH of the media and therefore eventually the overall cell viability of the culture. On the other hand, the internal levels of CO2 need to be maintained in order to ensure a proper physiochemical balance through different cell cycles. If either the external or the internal levels mess up, the result may see drastic changes. The internal levels are maintained through media and sometimes the cells themselves. However, the external carbon dioxide levels are maintained through your incubator. That is why it is important to know what is the carbon dioxide recovery, carbon dioxide maintenance levels of your incubator at all times. My colleague Carl has more insight on these topics that I just spoke about, and therefore I would like to pass over the rest of the presentation to him. Carl, over to you.
Thank you. So, as Nehal touched upon, we're going to look at really emphasize homeostasis within your cell culture, right? And so we can look at, in this slide here, you can look at a classic but important example of homeostasis as it pertains to cell cultures, and that's how cells maintain a proper pH, which is ideally sits around 7.8. So hyperacidity in a cell culture results in an irreversible loss of function of cells or impairment. And alkalinization also causes cell death. So you can't go too far either way, right? A couple of examples we have here, we can see that they stress both of these conditions. And alkalinization or hyperacidity in cell culture showed that, the increase or decrease in pH really resulted in not only cell death, but overall lack in productivity within a lab. So in the cell culture, we rely on these tools to provide a homeostatic environment. And in our case, we rely on an incubator. So this is done, of course, through buffering media, but also tightly controlling CO2 levels. So these demands are necessary, since frequent door openings and so on, CO2 levels are compromised very often in this environment. So we really have to have good tools and good technologies to bring CO2 consistency back into our tissue cultures. So we do this through sensors. In our case at Panasonic, we have kind of a next generation of CO2 sensors, and it's called an IR2 sensor. So maintaining proper CO2 levels and thus proper homeostatic cellular systems within an incubator really requires precise CO2 sensors. So in our sensor, an IR radiation, so infrared radiation, is directed to the sensor after passing through a filter. and a filter in front of the detector prevents wavelengths that are not specific to CO2 gas from passing through the detector. So from there, the light intensity is detected and converted into a gas concentration value. So every IR sensor relies on the principle that every gas will absorb at a specific frequency. In the case of CO2, it'll absorb light at 4.3 micrometers, which is the infrared band of the light spectrum. So to kind of prove the reliability and precision of our Panasonic sensors, we conducted a third-party study that compared incubators, with different types of sensors, so more traditional sensors that are used in CO2 incubators, and the cells in the Panasonic IR2 sensors actually had a higher percentage of viable cells across the 4-day study. So it kind of shows the delivery of a more precise environment using our sensors with CO2 recovery. So another example of how homeostasis plays a role in tissue culture is you can look at an O2 pathway or an O2 system of homeostasis. So as with carbon dioxide, external oxygen also plays an important role in biological systems and homeostasis. So here I just have a sort of a general diagram of some of the interconnections of oxygen and biological systems. So, more and more we're really seeing a demand for good oxygen control within stem cell or within tissue culture. And the good example here is stem cells. So as research goes on, more and more studies are really showing the importance of O2 control, oxygen control within the system that they're studying, and stem cells has been the front runner here. So recent studies have showed that the increasing importance of physiological oxygen in a stem cell. So if you kind of compare it to traditional just CO2 incubation, you can see that the benefits of culturing cells at physiological oxygen really bring it closer to in vivo levels. They have fewer chromosomal breaks, like the cells can be passaged longer, they're just more robust and healthier because lower CO2 levels as opposed to atmospheric CO2 levels will really mimic that in vivo environment. So it's a really good, again, tying into this idea of homeostasis and the tight control of oxygen is a good example here. So, another topic that's extremely hot in cell culture is obviously contamination control. Anybody who's done tissue culture is very familiar with this idea. So really we have to bring in a good technology to control contamination within our cell culture. So we use the example of mycoplasma because it's the most ubiquitous and widespread contaminant out there. Not only is it widespread, but it's actually really hard to see and to detect within your culture. So it doesn't really wipe out your cell cultures, but it can really affect your cells and your experiments in a number of ways. So mycoplasma contamination can be brought in a number of different ways, which is what makes it widespread. We can bring it in, a scientist can bring it in, like your staff can have it all across their bodies and arms and lab coats or whatever. They can be cross-contaminated from other cell lines. It can actually be brought in through protein sources or media sources, serum sources within your media, and all other equipment that's not really proper sterilized can also bring this in. So that has a number of effects, right? It can impair your cell growth, it can reduce metabolic activity, and modulate a lot of different reactions. So your cells aren't actually reacting how they should react, right? And the greater result of that is you can lose samples, but more so you can lose time. You'll have to spend more money on resources, and worst of all, you can have irregular or untrue results. So we have to address these types of things within our CO2 incubators, right? And that's become a demand in the market, right? So now when someone's buying a new CO2 incubator or they're outfitting a lab, they really stress the importance of contamination control. And that's been done in a number of ways. So a popular method is actually to do a high heat cycle within your incubator. So to take all of your cells and cultures out and just really ramp up your incubator to to extreme heat to denature any sort of cells or contaminants within there. Panasonic actually has taken a different route. So we have used two different methods to control contamination on a couple of different levels. We use hydrogen peroxide as a decontamination source, so we vaporize hydrogen peroxide within our chambers, and we also use ultraviolet light to resolve that hydrogen peroxide into harmless water droplets and O2. So this has a lot of benefits in the sense that it takes a lot less time to really fully decontaminate your cultures, and it's a lot gentler on your incubator system. So this diagram really quickly does show that H2O2 within an incubator, it's a novel technology, so it presents a lot of questions as far as its safety. But if you use the power of hydrogen peroxide combined with UV, your incubator and your cultures that go in after this decontamination cycle are not compromised here. So they're actually, once the cycle is done, you're just left with basically sterilized water in your incubator, and your turnaround time is a lot quicker. So here I've kind of just presented some just general metrics that show the precision, and then again the idea of homeostasis that ties back in to our incubators, right? And that goes with any sort of these environmental factors. We have really quick humidity recovery, really quick temperature recovery, and of course, as I mentioned, the decontamination time is a lot quicker. So this is all important, right? Because the cells need to maintain a homeostatic environment, and to do that, we really have to have a precise, environmental factors. So recovery after door openings or after you've actually used your incubator is something people don't really think about it because they kind of shut the door and just want to forget about it. But actually getting back to those levels, so getting back to the proper homeostasis, is really important. So again, using our Panasonic technology, we can really provide those environments very quickly. So then this diagram just kind of ties it all in as far as homeostasis. So if you have proper homeostasis, really what you're going to have is proper cell viability, and downstream from that, you're going to have more reliable and more repeatable experiments. So that can be factored in, as I talked about, with CO2 levels. And we use a really high-tech next-generation IR2 sensors to recover that. Talked about a little bit about oxygen levels, right? Physiological oxygen really plays a key role now, and we're seeing that a lot more and more in cell culture. And it actually ties into decontamination. The time spent decontaminating is a lot quicker, so we can kind of get back to the task at hand. So that sort of wraps it up. I'd like to thank everyone for letting us talk here today, and I'm happy to take any questions when the presentation is over.
Great. Thank you very much, Carl.
Thanks.
I think we'll go directly into our question and answer session. We do have a number of questions that have come in from the audience. And Peter, maybe I'll start with you. The spheroids that you were speaking of, how fragile are they, and how likely are they to be disrupted by pipetting?
The spheroids themselves are fairly robust. That being said, care needs to be taken. I know that some of the automated aspirating tubes can be quite powerful, and that can be of concern. But typically, we've had very good luck with manipulating the cells, essentially moving them from a hanging drop into a plate for assay, for example, and that's done by a quick centrifugation step, and they seem to hold their shape very well.
Great, thank you. Are there any ways to sort of promote cell aggregation, or are there cell lines that are particularly amenable to this technique?
There is some variability. I'm not an expert across all cell lines, but we do see differences. The MCF7 cells are fairly robust. They take 3 to 4 days, whereas some other cell lines, if you are trying to make, for instance, say a liver microtissue, then those can take up to 2 weeks to form. So it does depend on not only the cell type, but whether or not you have scaffolding. And of course, the addition of scaffolding can have some additional effects on how the cells might behave in the assay as well.
Great, thank you. Any other additions to that question for Kristina or Carl? No? Then I guess we'll move on. If you're considering automating or you're setting up a new cell culture lab, how important is the layout and the equipment placement? Do you need to consider for traffic and airflow and that sort of thing in your lab?
So a lot of times when you're laying out a cell culture system, an automated system, what's the most important thing to consider is obviously your space, but secondly, what your workflow is. A lot of times if you have a workflow where one of the processes you're just doing confluence imaging from the incubator to the reader, you don't want to have the plates go through the liquid handler for that because it may be able to do some other process at the same time in parallel while your incubator is handling the growth and then maybe a robotic arm taking plates from the incubator to the reader. So you definitely want to consider your workflow and how the instruments are set up around each other so that way you can optimize parallelism as well as timing and efficiency. In terms of airflow, it really depends on if you're going to have it in a clean room. We haven't really seen any effects of how the system's set up on clean rooms. Yes, in a hood, obviously you don't want too much equipment in there with covers on it. So a lot of times the liquid handlers have all the plexiglass covers removed if they're put into a biosafety level 2 cabinet. But those vendors that sell the biosafety level 2 cabinets always perform and certify the hoods after the liquid handler or the other devices have been put into the system for you.
Great, thank you. Any other additions to that question? No? Well, maybe Carl, I'll direct this one at you. You spoke about contamination in your lab and prevention. If you're dealing with a contamination in your lab, where do you start the cleanup? Do you start with your equipment or with your media? And how do you identify your source of contamination?
Well, I mean, you mentioned media. If you do have a contamination, it's really not worth your time to try to figure out where it's coming from because it's really your attempt is going to be futile. So really, I mean, if you're dealing with media, if you really know that you have contamination, I would really just start fresh as far as media is concerned. In the sense of cell culture, if you are reusing sort of any equipment, obviously that would have to be the start. Your little things like your pipette aids and that sort of thing would really have to be decontaminated in the best way so that it doesn't affect their functionality, obviously. And you have to take a look inside your laminar flow hood and that sort of thing, and just kind of cover all grounds where you're actually handling your cultures. But if you do have contamination and it's been confirmed, you really need to take a good look inside your incubator. So if your incubator does have a decontamination system built within it, I would run that. Do your hydrogen peroxide / UV cycle, or run your high heat decontamination just to kind of eliminate it. Because if you think about it, these contaminants they're living organisms just like your cell cultures, and they love the warm, dark, and humid environment within your incubator. So a lot of times, if you do have contamination, regardless of where it started, it'll really be harvested, and it'll grow to higher titers in your incubator because it's really just acting like an ideal environment for them. So that's why, we stress the point of really decontaminating the incubator because, the place I would focus. But obviously, you can't neglect the other places in the lab as well.
Great, thank you. Any additions to that, Peter or Kristina?
Yeah, I would just like to mention, on an automated system that includes an incubator, as Carl had mentioned, looking at what kind of decontamination options the incubator has, but also, on a liquid handler, if you're using liquid handling devices, make sure you look at what type of pipetting and how lab water is being manipulated around the instrument, how lids are being handled. Make sure that plates aren't being read in readers without the lids on them, stuff like that. Changing filters and good laboratory practices.
And I'll make another point. Thank you. That we kind of focus in on external or microbial interference as far as decontamination, but another big problem, and it ties into the proper aseptic techniques, is cross-contamination between cell lines. That's actually a big problem, and it goes less noticed. So you really have to be careful to run decontamination cycles, between different protocols, or not bring two different cell lines into a working space at once, because you have two different cell lines, you're really going to mess up the metabolism and the cell functionality of the quote unquote contaminated cell line. So that's also something you really have to keep in mind.
Great, thank you. So if you are bringing a new cell line into your lab, what protocols would you suggest to avoid cross-contamination?
Well, I think it depends on where your source is. A lot of times, you can sort of order in a cell line from a good source like ATCC. It's been confirmed that you're actually working with a cell line that's of interest. But if you, for example, get a cell line from a colleague or someone in the building that's just has been growing this using their own methods, and you just kind of take their word for it, I would really suggest, before really starting getting nosediving into a lot of really long-term experiments, to just confirm that the cell line is what it is. You can do that in a number of different ways. Sequencing would probably be the number one suggestion, just to make sure that it's really pure and that you're dealing with you have a good foundation. It's like having a good foundation for a house. You can build a mansion, but if the foundation is bad, then it's going to crumble. Also, there's a lot of you may have ordered in a cell line or brought in a new cell line. There's a good chance that could be contaminated, not necessarily with bacteria. With your common bacteria, but it can be contaminated with mycoplasma, which is actually considered a bacteria, but it's a lot more elusive, so to speak, that it really goes unnoticed under a microscope, or it doesn't actually wipe out your cell culture, so the cells aren't actually dead, but it's adhering to the cells and affecting their metabolism and their characteristics. So I would suggest number one, really confirming that you have the cell line of interest, and number two, doing mycoplasma tests. There's a number of different kits that you can do to just confirm that you don't have that contamination and that you're not bringing that into the lab.
Great, thank you. Kristina, this question is directed at you. What's the best method to keep cells from settling, and what's the minimum amount of cell suspension to have in the reservoir when plating in a 96 or 384 well plate?
Oh, yeah. So there's multiple ways that you can keep cells in suspension on an automated system. If you're doing a low throughput experiment, the use of multiple probe heads is nice in a trough to just do a quick aspirate dispense, maybe 2 to 3 times slowly to resuspend the cells, and then you can plate them. But for larger volumes, usually your source would be a trough on deck, but then you would have a continual supply of a cell suspension that would be pumped into this via proximity sensors or sensors on deck that would tell the system when the trough's getting full or empty of cell suspension, and you could take more out of your source container. So you can handle as much as you want, really, depending on what your lab requirements are. So flasks below the instrument or to the side of the instrument that are on a hot plate, say, with the stir bar keeping the cells in suspension. And then they can be pumped onto the system deck and then put into a little module like I showed that keeps them continually in suspension on the instrument platform while you're going through your plates of plating cells or what have you. So it really depends on throughput. Yeah.
Yeah. Carl, I'll direct this next question at you. How frequently should you be calibrating your CO2 sensor and temperature controls on your incubator?
That is a great question. Absolutely. If you have a new incubator coming into a lab, 100% have it calibrated. A lot of times your sales rep or the technician that's actually setting up the incubator will do that. But you should really kind of just confirm that that is calibrated with CO2 and temp especially. So I would say, to be safe, especially if you have a GMP or a really highly regulated system, to get them calibrated once a month, I think it's worthy for labs to invest in, any sort of calibration equipment to see, that they're reading the correct temperatures. You'd be surprised at how many times, technicians will walk into a lab and the CO2 is reading at 3.5 when the scientist thinks it's reading at 5. So really, scientists really trust what's being read right on your screen, but that's not necessarily true. So I'm glad you brought that point up because calibrating your incubator is extremely important, and you can avoid a lot of issues for doing that. So 12 times a year, once a month is probably good.
Great, thank you.
Mm-hmm.
Any other comments there? No. Peter, I'll direct this question at you. How good is the technology that you mentioned for culturing neurons and neuroglials for long-term use?
We have done some work with neurons as far as tissue culture. I'm not sure. One of my colleagues been doing some work with that, and I'm not sure to what extent he has automated that yet. We have not done a lot of high throughput screening. However, the cells from perspective of cell culture and cell seeding. Typically, the cells that we've used, all the cell lines that we've used, have performed fine. Typically, we'll use the peristaltic pumps, as I mentioned, because the fluid path is autoclavable. They're cassettes that can be calibrated and autoclaved repeatedly. And we have not really had seen any detrimental effects of any cell lines that have gone through the peri pumps as far as seeding. The Citation 3 has environmental controls, which include CO2, temperature, and nitrogen. So if that's necessary, and so those are certainly helpful for long-term or longer-term imaging experiments or other optical modes of analysis in the reader. Humidity control is something that needs to be considered for longer-term experiments that are done in the imager itself, and there are different types of plates that we've tried that have reservoirs for liquids, very much like the hanging drop plates. My initial comments, the thoughts would be they should be fine, but without further optimization and testing of that particular cell line. I cannot say definitely how long or what length of assay can be run without essentially taking that plate and putting it back in an incubator, which is what we'll do a lot of times as well, very much like an automated system where the plate would go into the imager, be read, and then taken offline and incubated for a day or so. But it sounds like this question is more directed to, can you put the plate in, image neurite outgrowth or something of that sort over a day or two. And I think the system should be suitable for that, and we're hoping to actually do some additional experiments here in the near future showing exactly that.
Great, thank you. I think we have time for one more question here, and I'll throw this one out. You can all take a shot at it. What's missing from the researchers' toolkit right now, and what do you expect to see on the horizon for cell culture equipment?
I mean, I can add a piece to this. I really, as I kind of touched upon in our presentation, the equipment is out there, but I think a trend in cell culture and general cell culture will be really to drive the physiological oxygen idea. So, there's a lot of different methods you can go about doing this. You can have tri-gas incubation. You can actually dissolve chemicals like cobalt chloride into your media to kind of give it a hypoxic or more of a hypoxic environment. But I think really getting this technology down to make it standard, to have a lower physiological oxygen level is really what's missing, from our end in the cell culture world.
I can add to that too, as a trend more than what's missing. Yeah, as Carl mentioned, the tools are there. I think just finding an efficient and effective way to, from my perspective, integrate them together so that a researcher can, perform a whole entire cell culture process or cell-based assay process in an automated fashion so that they can increase their throughput. And also handling, volume size, so large volumes or small volumes, having equipment to be able to do both and flexible for both, as well as the software that the system uses to be able to handle these runs and seamlessly integrate all these components together and maintain a clean, sterile culture cell-based assay system that can run for weeks.
Thank you. Peter, any parting words?
No, I agree with what both Carl and Kristina said. Additionally, maybe some smaller footprint instruments. I think those are, kind of gearing towards the academic lab. We're seeing some more interest in some of these cell-based assays moving into the academic labs, where cost becomes more important there as well. So, smaller footprints. And smaller automation systems, I think, are going to become more trendy as well.
Great, thank you. Well, thank you for your discussions today, and this brings us to the end of today's webinar. If you have any further questions, please consider reaching out to the panelists directly, as they are the true experts in this field. Their contact information, as you can see, is available on the screen now. Just a reminder that today's webinar will be available at the link you see on the screen now. On behalf of Lab Manager, I'd like to thank our panelists today for all their hard work they put into their presentations, and I'd like to thank our sponsors, BioTek Instruments, Panasonic Healthcare Corporation, and Hamilton Robotics for supporting this webinar. As well, I'd like to thank all of you for taking time out of your busy schedules to join us today. Please mark your calendar for our next Tech Trends webinar, which will focus on how an electronic lab notebook can benefit your lab. This webinar will be taking place Thursday, July 17th from noon until 1:30 p.m. Eastern Daylight Time. For more information, please visit our website at www.labmanager.com. We hope you can join us again next time. Thank you and have a great day.